Hi, everyone. My name is Chris Smith. I'm a PhD candidate in the Department of Anthropology at the Graduate Center of the City University of New York. And today I'll be talking about the importance of the otolith system in human cognition and talking a little bit about my dissertation work along the way. So the otolith system is part of our vestibular or balance system, and the organs responsible for this sense are housed inside the labyrinth of the inner ear and the petrous portion of the temporal bone. Here, I've highlighted the vestibular part of the labyrinth, and I want to draw your attention specifically to this area underneath the canals called the bony vestibule. It's inside this bony vestibule where you can find the otolith system. So while the semicircular canals detect angular acceleration of the head, the otolith system, comprised of two otolith organs, the utricle and the saccule, instead detect linear acceleration and head tilt relative to gravity. While the utricle detects horizontal movements, the saccule instead detects vertical movements. So growing evidence is suggesting that this otolith system provides an essential gravitational frame of reference for cognitive processes that are fundamental to our concept of the human mind. These include things like bodily self-consciousness, spatial memory, and even social cognition. By providing such a framework, the otolith system is foundational for interpreting sensory signals in 3D space and facilitates signal integration with other cognitive processes. The aim of this talk is to review some aspects of differences in brain and vestibular structure that I've come across in my dissertation work, which may be associated in extant hominoids. This may suggest the importance of studying the otolith system in order to better understand human cognitive evolution. I'll now discuss two examples where I've observed this difference in brain and vestibular morphology. First, talking about the cerebellum and bony vestibule. So recent evidence from Barton and Venditti in 2014 primarily is showing an expansion of the cerebellum relative to the neocortex in hominoids compared to other anthropoids. And we know functionally that the cerebellum plays a vital role in the vestibular system as a whole, since the vestibular end organs send signals either directly or indirectly to certain regions of the cerebellum. If we move now to talk about the bony vestibule itself, published data combined with results from our own lab point to an expansion of this bony vestibule in hominids, along with a structural reorganization, with the greatest expansion in volume found in Homo sapiens. Here you can see a comparison of the expanded bony vestibule in an orangutan compared to a macaque. The area of the bony vestibule here is highlighted in purple. This could potentially reflect differential otolith function, and it's interesting that many of the same taxa exhibit similar changes to both bony vestibule and cerebellar volume. Going a bit further, here's a principal components analysis plot from our ongoing work to be presented in the spring, showing differences in some aspects of vestibule structure related to the utricle and saccule, where modern humans exhibit a fairly substantial difference in the bony structure of this region compared to great apes, which are all clustering together on the positive end of PC1. This difference is mainly related to the bony structure around each otolith organ and the configuration and positioning of each. Another specific example is the relationship between the hippocampus and saccule. We know the hippocampus and saccule specifically are known to be associated with a recent study linking a loss of saccular function to decreased hippocampus volume in humans. Comparatively, we may also see evidence of this association in the species Hylobates lar. It's been noted in literature that Hylobates lar exhibits a relatively expanded volume of the hippocampus compared to other hominoids. My preliminary observations of Hylobates also suggest the spherical recess, shown here highlighted in blue on the bony labyrinths, is also enlarged relative to the rest of the bony vestibule. Here you can see a comparison between Hylobates lar and Congo pygmaeus. This is intriguing as both the saccule and hippocampus both play an essential role in spatial memory. So we propose that these observations illustrate the potential connection between observable morphological differences in the otolith system and brain structure. Because of this, the otolith system may provide an additional approach to examine cognitive evolution of the fossil record, especially in human evolution. It could also provide an alternative approach to investigate other cognitive behaviors that may be difficult to interpret in the fossil record, like spatial navigation. And with that, I just wanted to say a small thank you to all of my advisors and mentors who have been involved with this research, and also a thank you to the Fossil to Minds Workshop Organizing Committee for allowing me to talk today. Thank you so much.